See the slide? Okay, today we shall discuss about DNA topoisomerases and their function. What are the topoisomerases? Topoisomerases, as we have discussed in the previous lectures, that DNA undergoes the coiling, and that is positive supercoiling or negative supercoiling. Naturally, DNA is meant for the coiling as a result of binding of certain histone proteins
स्लाइड
and
शो
various. Okay?
स्लाइड शो
Can you
नहीं हो रही
can you see the slide now?
नो सर
Okay. Can you see the slide now?
यस सर
Okay, so as I have told you that DNA, there are negatively occurring coiling in the DNA that are either negative supercoiling or positive supercoiling. So DNA supercoiling is controlled by a group of the enzymes. They are naturally present, and those enzymes are called DNA topoisomerases, or simply we can also call them as topoisomerases. So topoisomerases are basically the enzymes which control the supercoiling in the DNA. Two classes of the topoisomerases are there. One of them is type 1 topoisomerases, and as we can see here, this is the schematic mechanism of action of the topoisomerases 1. So here are some characteristics of topoisomerases 1. This was first time reported by James Wang in 1971. It basically act by creating single strand break in the DNA. Here you can see that this topoisomerase, it will induce a single strand break in the DNA, and it will utilize the energy. So topoisomerases 1, they basically utilize the energy, and type 1 topoisomerases 1, they are always denoted by Roman odd Roman numerals. Mean topoisomerase 1, 3, 5, they will be odd Roman numerals. We will always denote them. There are subclasses of these topoisomerases 1, for example 1A, 1B, and 1C. These are the sub classes, and these subclasses are based on the amino acid sequences, sorry, on the basis of the sequences. Amino acid sequences are mechanism of their action. So here is the schematic mechanism of the action of the topoisomerase 1. The enzyme cut a single strand of the DNA duplex, pass the uncut strand through that. In the second step, in the first step, it will induce a single strand break in the double stranded DNA, which is linked or which is coiled, and in the second step, it will pass this uncut strand through this cut region. This uncut strand will be passed through this break, and ultimately the ligase enzyme will ligate, and hence we will have an intact, what we can call it as, we will have an intact type of the DNA there. So this is type 1 topoisomerase. When we talk about the type 2 topoisomerase, Martin Gallett's 1976 first time discovered these topoisomerases. They induce the transient double strand break in DNA. They also utilize the energy in the form of the ATP. They use the ATP for their function. And but the difference of the function between the type 1 topoisomerases and type 2 topoisomerases is that type 2 topoisomerases, they induce the double strand break in the DNA, and they are denoted by even Roman numeral. For example, 2, 4, 6, 8, the type 2 topoisomerases, they will be denoted. Their subclasses are 2A and 2B, and their subclasses are, their classification is based upon their amino acid sequence. On their amino acid sequence, on the basis of their amino acid sequence, we can classify them. So if we further elaborate the type 1 topoisomerases, type 1 is, there are further subclasses of type 1, as all of you know, that type 1a, type 1b, and type 1c. Type 1a topoisomerases, they are present in all cells. They only relax the negative supercoiled DNA. There are two types of the supercoiling. You can study from the previous lecture. I will share it with you that sometimes the DNA is negatively supercoiled and sometimes the DNA is positively supercoiled. So when there is negative supercoiling, type 1a topoisomerases, they will relax that negatively supercoiled DNA. It can also be present in E. coli. Topoisomerase 1, it is having omega protein subunit. Topoisomerase is 3. This is also included in the type 1a. Similarly, it can be present in U bacteria and archaea. It also involves the reverse gyrase enzyme, strand passage mechanism, as you have seen in the previous slide, and it form 5 from 5 prime to third prime, or from 5 prime to any type of the phenylalanine tyrosine intermediate. This is their amino acid composition. When we talk about the type 1b topoisomerases, they are subclass of the type 1 topoisomerases. It is present in eukaryotes and many prokaryotes. For example, E. coli, it can relax both type of the supercoiling. Either it can relax negative supercoiling as well as positive supercoiling. It is operative in humans as well. Topoisomerase 1 is included in type 1b. It control the rotation mechanism, and it form the third prime and phenylalanine tyrosine intermediates. We have type 1c. It is present in archaea, and archaea is also having topoisomerase 4. Similarly, it control the rotation mechanism, and it form the third prime phenylalanine tyrosine intermediate. Some hyperthermophiles, for example, U bacteria and archaea, they also have this type of the topoisomerases, which are called topoisomerase 1. Type 1a topoisomerases, sometimes they are also called as reverse gyrase. They induce positive supercoiling in DNA, so the ATP driven action. Mean it utilize the ATP, and this gyrase or reverse gyrase, it will induce the positive supercoiling. Sometimes another type of the CF gyrase, another type of the gyrase, it can induce the negative supercoiling as well. So positive supercoiling will tightens the DNA double helix, and it will protect the DNA from thermal denaturation. Mean when DNA is positively supercoiled, it means it will be tightened to its complementary strand, and there will be no dangers or no damage from the heat or from the high temperature. So enzyme, as we have discussed, that it basically cuts one of the two strand. Here one strand, this is moving from 5 prime to 3 prime direction. The second strand is moving from 5 prime to 3 prime direction. So apparently this enzyme cuts a single strand. Here at this central hole of this enzyme, this is whole topoisomerase 1, and it will induce a single strand cut, passing a single strand loop through the resulting gap and then resealing the break, thereby twisting the double helical DNA by 1 turn. So as a result of which, now when there is cut, it will pass this uh, greenish strand through this red strand. So there will be addition of 1 turn. 1 turn will be added. Type 1 topoisomerase function via strand passage mechanism. Mean initially it will induce a cut. In the second step, this strand will pass through this strand, and here a turn will be developed there. So the protein recognizes a single strand region of the DNA. For example, this is red strand. This is recognized by the protein, and it will bind to this area and bind it in its binding groove. It will. This is the yellow area is representing the binding groove of this protein or this topoisomerase, and this topoisomerase via its binding groove will bind one of the two strands of the DNA. I mean, it will bind to the red strand, or single strand, which is denoted by red color. This is followed by opening of the gap between domains 1 and 3. As you can see here, that these are the domains. This is domain 1 and this is domain 3, and in this example here, you can see that the gap is still closed. But when this binding groove has binded with the one strand of the double-stranded DNA, there will be the opening. This first step is binding. In the second step, there will be opening up between the domain 1 and domain 3. Domain as the region of the protein or the enzyme. Enzyme, as all of you know, that most of the time they are protein in nature. So domain is the region of the protein by which it binds with the DNA. Two terminologies are used. One is motif and other is domain. So domain is the region of the protein by which it binds with the DNA. So single-stranded DNA, which is B-like DNA conformation, it will be its other strand will be excluded from the groove. Here you can see that this greenish strand is completely excluded from the groove, whereas the red strand, it is completely bounded by the binding groove of this topoisomerase 1. So DNA is cleaved when there is opening. As here you can see that the gap has been opened between the domain 1 and domain 3, and the DNA is cleaved. So newly formed 5' end becoming covalently linked to the active site tyrosine. So here you can see that now this is 5' to 3' and this is 3' to 5' because of the link. So this DNA will bind to the tyrosine amino acid which are present on the surface of this protein, because this is enzyme and it is proteinaceous in nature. So the end of the DNA which is cut by the binding groove of the topoisomerases 1 end will bind to the tyrosine amino acid of this enzyme or this topoisomerase. This is intermediate tyrosine 5' intermediate phenylalanine tyrosine link will be formed. Newly formed 3' end remaining tightly but non-covalently bound in the binding site. This is now we have two newly formed end. This is newly formed 5' end and this is newly formed 3' end. So this will not bound to any other protein or any other groove or any other region of this topoisomerase 1. It will remain in unbind form, whereas the newly formed 5' end will bind to the tyrosine amino acid of this topoisomerase 1. So unbroken, which is called green strand, is passed through the opening or gate formed by the cleaved red strand. Now this gap will be filled by the passage of, as here you can see that the 5' free end, it is not actually free, it is bounded with the tyrosine of this topoisomerases, and 3' newly formed 3' end is free. So the gap will be filled by the passage of this green strand. It will pass through the red strand, and it will also enter into the protein central hole or the central channels. It will also enter into the protein central hole. The unbroken strand is trapped by the partial closing of the gap. Now the gap will start to close. The two cleaved end of the red strands are rejoined. When the gap is closed, this strand it will detach itself. It will detach. This is what we can call it as strand entrapment. The strand will be tightened by this hole, by this channel. Now the two strand or the two end of the red strand, they will again rejoin with each other. This will be freed. This will detach itself from this topoisomerase. It is already free, and these two ends they will be rejoined, and as a result of which cleavage reaction will be reversed. So that's how this is religate, mean these two strand, these two end of this red strand, they will religate. Enzyme ligase will again rejoin them. So this is whole mechanism of the action, that initially the topoisomerases, it will bind to one of the two strands of the double-stranded DNA via its binding groove, and it will result in cleavage of the one strand, and the two free end of this cleaved strand will be produced. One end will bind to the tyrosine of this enzyme, and other will remain in the unbinding form. Then the third step is strand passage. The intact strand or the green strand, they will pass through the central hole of these topoisomerases, and the force now strand entrapment mean it will be tightened by this gap. Now here you can see that the gap has tightened. Here you can see there is opening of the channel. This is domain 1, domain 2 and domain 3. The gap will increase. Now after the entrapment, the domain 1 and 3, they will be very much adjacent to each other. The gap will be tightened now, and ultimately the two end of this cut strand, they will be religate, and hence there will be passage. There is formation of 1 ton. 1 ton will be increased. So it means the topoisomerases, they can induce the coiling. So when there is increase, one ton has increased, it means the coiling, supercoiling has increased. They can increase or decrease the process of DNA supercoiling. So they can be operative in both ways. They are operative in both ways. Here you can also see the type 1b topoisomerase function via a controlled rotation mechanism. Topoisomerase 1 relax DNA supercoils by permitting the cleaved duplex DNA loosely held downstream segment to rotate relative to the tightly held upstream segment. So here you can see that first topoisomerase will bind and it will melt, and when there is melting, here you can see that there is rotation. Now the strand has rotated, this has moved in this direction loosely, loose rotation, and the other has moved in this direction, and this break will rotate in the second step, and here you can see that there is joining, rotate and will rejoin as a result of which, and this rotation can only occur above the sugar phosphate bond in the uncleaved strand, mean alpha, beta, and epsilon and eta, I guess the last one. These are opposite to the cleavage side because of the cleavage freeze these bonds to rotate. Mean they can easily rotate because of the sugar phosphate bond. As all of you know that, please unmute your mic, mute your mic, because as all of you know that sugar and phosphate, there is bond between the sugar and phosphate, that is called phosphodiester bond. So here is mechanism of the action of type 1b, that type 1b first bind to the double stranded DNA, it will melt and it will induce a break. So the break has been induced at this point. Now it will pass this intact strand will be passed through there, and ultimately we will have the this is pass join mechanism, and in the second process there is rotate join mechanism. In this case, this will bind, this topoisomerase 1b will bind, it will break. First thing, it will break, and it will also induce the rotation. It will be rotated in this direction. So when it is rotated in this direction, so here you can see that it has bind to the different strand. It has bind to the red strand, whereas in this case it has bind to the green strand, and it will result in the rotation of this strand. I mean, it will rotate in this direction, and it will rotate in this direction, and ultimately these rotated strand they will rejoin. So this is all about the type 1 topoisomerases. When we talk about the type 2 topoisomerases, as all of you know that they are further classified as type 2a and type 2b. Type 2a is present in bacteria and in the form of A2B2, which is heterotetramers, DNA gyrase or just gyrase. It can also be called. Sometimes topoisomerase 4 is also present in type 2 a subclass. यूकरियाट में ये ए बी होमोडाइमर की फॉर्म में है, ठीक है ना. टोपोआइसोमरेज 2 जो है ये भी इसमें जो है वो इंक्लूड है. टाइप 2b में अगर हम देखें तो only one member that is present in archaea. Sometime topoisomerase 6 that can also be included in the this type 2b. So topoisomerases, they sometime unknot or disentangle the DNA molecule. In addition to the relaxing the supercoiled DNA, topoisomerases sometimes they also promote or they also perform several other important functions to maintain the proper DNA structure. For example, they can use the same DNA break and strand passage reaction to relax the DNA to carry out the reaction. As all of you know that DNA should be relaxed to express the genes during the process of transcription and translation. Topoisomerases can catenate and decatenate the circular DNA molecule. As all of you know that when we talk about the prokaryotes, they are having the circular DNA, and sometimes that circular DNA is entangled with each other. That is called catenation. So to remove those entangled DNA, the topoisomerases, they also perform their role, that is called decatenation. Here in this example you can see that how topoisomerases, they can unknot and disentangle the DNA molecule. In the first case, type 2 topoisomerases, these are two strands, they are entangled with each other, this is called catenation, and when they are freed from each other, this is called decatenation. So topoisomerases, type 2 topoisomerases, they are involved in catenation and decatenation. They covalently close circular DNA. This is called cDNA, triple cDNA, covalently closed circular DNA. So when they are in this configuration, this is called catenated DNA, and when they are in this free condition, they will be called as decatenated DNA. Similarly, type 1 topoisomerases, they can only catenate and decatenate molecule if one DNA strand has a nick or the gap. So type 1 topoisomerases only decatenate if one strand, one circular DNA is having a nick or gap. At this point, it can decatenate, or it can free this black circular DNA from the red one. But if there is no nick or break, the type 1 topoisomerase will not be able to decatenate this DNA. In the C diagram, here you can see that entangled linear DNA molecule during the replication of eukaryotic chromosome. This entangled DNA, they can be arranged in the linear configuration. And in the last diagram here, this is called DNA knotting. Sometimes the DNA can also be present in this form, which is called knotted DNA. So this knotted DNA can be unknot by the topoisomerase action. Topoisomerase can also unknot this DNA, and it will assume the relaxed shape. So topoisomerase, basically how they cut the DNA and how they will rejoin the DNA. As I have told you that topoisomerase will always operate in 5' to 3' direction, as 5' end of the DNA is bounded by the by the phosphodiester bond. So here the topoisomerase, it will cut the DNA at its 3' end. So now we will have two free end. This is 3' free end, and this is 5' free end. And at this 5' free end, again the topoisomerase will use itself, and it will again rejoin this end after removing the knot, or after inducing the what we can call it as coiling or supercoiling. It will again rejoin. So topoisomerase is basically first it cleaves the DNA. Only a single-stranded DNA is shown here. Single-stranded DNA, we have shown that same mechanism is used by the type 2 topoisomerases, and in that particular case, two enzyme subunits are involved. So here you can see that in the first instant, the topoisomerase will attack on the 3' end of this DNA, and it will induce a break, as a result of which 3' to 5' one end will be produced, and 5' to 3' other end will be produced. It will bind itself to the 5' phosphate group. Where phosphate group is added, the topoisomerases will join there. It will break the phosphodiester bond, and it will join itself there. When the break has been induced, and now either the strand passage or any other type of the coiling mechanism has been completed, after that this 3' again will attack to this OH will again attack to this region, and as a result of which now this phosphate will again rejoin with this oxygen. Phosphodiester bond will be formed, the strand will be rejoined, and this topoisomerase will be freed after rejoining. So this is the mechanism of the action of these topoisomerases. If we talk about the role of the topoisomerases, topoisomerases during the DNA replication and transcription, sometimes the DNA becomes overbounded, or it is having high supercoiling. So that supercoiling ahead of the replication fault. फॉर एग्जांपल आप किसी भी जो है वो रस्सी को या तस्वीर को जब आप टाइटन करते हैं तो जहाँ से आप उसको खोलने की कोशिश करते हैं उससे आगे आगे आप देखते हैं कि उसमें जो है वो फर्दर जो है वो कॉइलिंग जो है वो शुरू हो जाती है। तो जैसे रेप्लिकेशन फॉर्म में आगे हम देखेंगे जहाँ से डीएनए एक दूसरे से अलग होता है, फ्री होता है, तो उसके दोनों तरफ जो है वो एट द टू adjacent end जो है on the both side DNA is having overbounding. So if left unbaited, this torsion will eventually stop the ability of the RNA and DNA polymerase involved in the process to continue the replication process. So DNA replication process will be halted, will be affected if that overbinding is not overcome. So topoisomerases, they bind and they to the either to the single strand or the double strand DNA and cut the phosphate backbone, as we have previously discussed. An intermediate break will allow the DNA to be untangled or unwound from each other. At the end of the process, the DNA backbone that is resealed by these topoisomerases. So winding problem of the DNA so this is I guess same. To remove the DNA supercoil during transcription and DNA replication. This is the function of the topoisomerases. Similarly, for strand breakage during the recombination, when one strand is crossover with the other, and that strand needs to be to break, that break is actually induced by these topoisomerases. So chromosomal condensation is also carried out by these topoisomerases. Similarly, they disentangle intertwined DNA during the mitosis. During the mitosis phase, the DNA that has been intertwined, that is disentangled by these topoisomerases. Okay जी, any question? Clear है? You have any question? Can you hear me? If you have any question, you can ask, please. Jay, if you have any question, you can ask, please. No question? Okay. At present, we have all the participants are there. Okay, stay connected. Let me download your attendance and